Everyone, this is Sheetal here. Today we're going to talk about gastric digestion. So first, let's talk about what is digestion. So digestion is nothing but the process of breaking down food into simple substances that could be easily absorbed into the bloodstream. This is generally done in two ways: mechanical digestion and chemical digestion. So mechanical digestion is nothing but manual breakdown of food, so that it could be easy for the enzymes to act upon it. And so that's mechanical digestion, and this starts with when we chew our food. So that's like part of like mechanical digestion. And similarly, in the other parts of the GI tract, there's something called the musculus externa layer, which is nothing but a three layer of like smooth muscle. And the contraction of this muscular layer causes the food to be broken down. And there is chemical digestion. Chemical digestion is nothing but degradation of the molecular structure of the food by digestive enzymes. So then now, since we're talking about like gastric digestion, let's talk about the stomach. The stomach is a J-shaped bag, and this contains like the stomach contains gastric glands that secrete gastric juice. So the gastric glands are generally divided into like three. That's pyloric, cardiac, and oxyntic glands, and they are tubular glands. So the cardiac and the pyloric glands are predominantly secrete or like secrete mucus, and this mucus is mainly helpful in protection of the stomach, like the lining of the stomach against the action of acid, and prevent self digestion of the stomach. So now here we see the picture of oxyntic glands and pyloric glands. So the oxyntic glands, like the first one here, is surface mucus cells. So these surface mucus cells generally produce mucus and bicarbonates. Now the mucus is mainly involved in like production of the lining of the stomach, and then bicarbonates are also there for neutralization of the acid, like again to protect the epithelial lining. And then we have the parietal cells. The parietal cells are mainly involved in secretion of HCl, as well as like intrinsic factor, which is mainly important for absorption of vitamin B12 in the terminal ileum. Then we have mucous neck cells, which again secrete mucus. And then there is enterochromaffin like cells, which is like histamine producing cells. So this histamine is mainly involved in stimulation of HCl from the parietal cells. And then we have somatostatin. Somatostatin is has an inhibitory effect, and like this inhibits HCl production. Then we have chief cells. These chief cells are mainly involved in production or production of pepsinogen, which is nothing but an enzyme. It's like a proteolytic enzyme for digestion of proteins, and the chief cells also secrete gastric lipase. So that's basically like oxyntic gland. And then we have the pyloric glands. The pyloric glands again have the surface mucus cells and mucous neck cells. G cells produce gastrin, which again stimulates HCl production. And there is also D cells for somatostatin. So this is just all I said before. And yeah, so when we talk about like parietal cells now, so parietal cells are like, as we know before, it's like for secretion of hydrochloric acid, and it's a pretty concentrated acid, and the pH of the acid is like zero point 0.8. And apart from that, it also involves in like secretion of intrinsic factor. And this is necessary for absorption of vitamin B12 in the terminal ileum. Then we're going to talk about chief cells. This chief cells, as we know, it secretes zymogen pepsinogen. So this is an inactive enzyme. So this is activated in an acidic pH. So when HCl is secreted in the stomach, it causes an acidic environment, and this acidic environment, which is this acidic environment activates like pepsin, and the acid level is generally supposed to be below 3.5 pH. So how does pepsin act? So pepsin breaks the internal peptide bonds of proteins in a pH of like 2 to 3, and converts the proteins into like peptides or like polypeptides. That then there is also gastric lipase, which breaks down fats. So then, yeah. So now let's discuss about the functions of HCl. So HCl, the main functions of HCl is are to create a hostile environment for pathogenic microorganisms that are like that are taken in through the mouth. Then it's also there for denaturation of proteins and make them more like accessible for enzymatic degradation by pepsin. And obviously, the third one is to activate pepsinogen. Then the barrier, we're going to talk about the barrier protecting the stomach wall against HCl. So this is really important because if the barrier is like broken or like if there is a some kind of a break in the barrier, then in such kind of a condition, there could be ulcers formed, like the peptic ulcers or gastric ulcers are because of this condition. So the barrier is really important. So the first one is the mucus coating of the epithelial surface, then the lipid content in the cell membranes, then the tight junctions near the luminal tips of the epithelial cells, which prevent diffusion of acid in between the cells, and the bicarbonate ions secreted by the surface epithelial cells. This is there for neutralization of the acids. And then now we're going to talk about the phases of gastric secretion. So there are three phases. Like the gastric secretion is secreted in like three phases. The first one is cephalic, gastric, and the intestinal phase. The phases of gastric secretion may overlap, and so the first one, cephalic phase. So the cephalic phase of gastric secretion occurs when there's a stimulus, like suppose like when we taste the food or when we smell the food or on sight and sound of the food, like we hear about the food. This phase of gastric secretion is like entirely reflex and is mediated by the 10th cranial nerve, the vagus nerve. Then we have the gastric phase. The gastric phase is again mediated by the vagus nerve, the local enteric reflexes, and by the release of gastrin, the hormone gastrin. So the gastric phase of secretion accounts for like 70% of the total gastric secretion. It's about like 1.5 liters. And this phase continues until the food has left the stomach. Then we have the intestinal phase. The intestinal phase is present when the food is present in the upper portion of the small intestine, like in the duodenum, and this will continue to cause like gastric secretions. So a small amount of gastric juice is produced in this phase, and this could be probably due to a release of small amount of gastrin in the by the duodenal mucosa. So then we are going to talk about inhibition of gastric secretion. Although like chyme slightly stimulates gastric secretion in the early intestinal phase, but then later it starts inhibiting gastric secretion. So this is mainly due to two. This is mainly because of two things. One is the reverse enterogastric reflex, and the other one is because of hormones like secretin, gastric inhibitory peptide, vasoactive intestinal peptide, somatostatin, and cholecystokinin. Then we're going to talk about like absorption and emptying. So stomach absorbs few of the products of digestion, like very few of the products. It can absorb small amounts of glucose and other simple sugars and amino acids and some fat soluble substances, like very few in very few amounts. And it also absorbs small amount of water, alcohol, and drugs like aspirin. The rate of emptying of the stomach depends upon the physical and chemical composition of the meal. Fluids empty faster than solids, carbohydrates more rapidly than proteins, and proteins more rapidly than fats. I guess that's the end of the video. Thank you for listening.